To shed some light on this wide-ranging topic, I am joined by Dr. Jeffrey Tomkins via Skype from his office in Dallas. Dr. Tomkins has a PhD in genetics, as well as a master's in plant science and a bachelor's in agriculture. While working as a faculty member in the Department of Genetics and Biochemistry at Clemson University for over a decade, he performed genomics research in plants, animals, and microbial genomes.
And anyways, this fusion site consisted of these telomere sequences, but it was very small. Human telomeres contain 10,000 to 15,000 bases of this sequence. So in other words, if you were to find such a fusion, you would expect something about 20,000 to 30,000 bases in length. But this fusion site was only about 800 bases in length, and it was only about 70% similar to what would be a pristine fusion. Yeah. So it was highly degraded.
Because they found these alleged fusion sites all over the place in other chromosomes, right?
Well, it turns out that internal telomere sequences are found all over the human genome. And there's actually been papers published on this in the past few years. And it turns out many of these sites are performing functional things in the genome. They're binding to things called transcription factors, which initiate a gene or cause a gene to begin to be copied into RNA. And as it turns out, in my research, the fusion site is actually read in what's called the reverse complement, and it's a transcription factor binding site in the middle of a gene called DDX11L2, which is an RNA helicase long non-coding RNA gene. So it doesn't code for a protein, it codes for a functional RNA molecule that's active in the genome. And it turns out this gene is highly co-expressed with other genes that are highly critical to the cell cycle and signaling in what's called the extracellular matrix, or the area outside your cells where a lot of signaling and communications go on. And it turns out this gene is highly expressed in many different cells in the human body. So it's a very important gene, and it's linked to many other highly important genes as well.
So this alleged fusion site is in the middle of this life-supporting gene.
Right.
The gene crosses over the fusion site.
Yes, across it. Actually, this site is in the middle of the gene. It's in what's called the first intron. So these genes, these long non-coding RNA genes, they function exactly like protein coding genes. They have exons or coding regions that end up in the final RNA molecule. They have introns, which are intervening regions that do not end up in the final RNA molecule. They have promoter regions and control regions exactly like protein coding genes. And in fact, your genome contains over twice as many long non-coding RNA genes as it does protein coding genes.
Wow.
And they're finding out now that that these things are critical for human health, growth, development. They're finding out that when many of these long non-coding RNA genes are mutated, that it leads to cancer, that it leads to developmental problems. And so this is actually the hottest area of research right now at the NIH, and their funding mechanisms is these long non-coding RNA genes.
So in our alleged ape ancestor, this chromosome would break into two, breaking this critical gene in two, so the ape couldn't live.
Well, what I'm saying is that you don't form new, highly functional, critical genes by slamming together two chromosomes. And
yes, that's a much better way to put it.
And what's really interesting is that there was a research group in 2002 that sequenced 600,000 bases of DNA surrounding this gene. And they actually reported that the fusion site was in what they called a pseudogene at the time, which is the gene I'm talking about now, which we now know 12 years later is highly functional and critical to the human genome. But they actually reported this back in 2002, but no one was really talking about it. But they sequenced 600,000 bases of DNA surrounding this gene and found that it was in a gene-rich region. It was in a nest of other genes, and that none of these genes in this entire 600,000 bases are found in chimpanzee.
Now that's a crucial
So
point.
they hypothesized, yes, and of course my research showed that as well, but they hypothesized that all these genes got there by coming from other places in the human genome. They didn't come from chimpanzee. This is the evolutionary thinking, the mindset. They didn't come from chimpanzee because there was no comparison to make. So they just came from other places in the human genome. So they just somehow ended up there through different mechanisms, supposedly. But
isn't that a convenient story?
yes, right. It's not one thing; it's another. As far as
So, what would happen to me if this gene were removed?
Well, it's difficult to study because this gene is not in primates. It's not in mice. And see, these are our model systems, especially the mouse model system is very important to studying human health and disease, because if that same gene is in mouse, then we can knock out that gene and see what the effects are. But you can't really do that in humans, or at least talk about it if you did. But anyways, when we study human genes, we have to do it in a system like mouse, where we can knock out the gene and mess it up and then see what happens. And so there's a lot of genes in humans that are not shared by other creatures, especially lab study creatures, and we just really don't know what they do. We can associate those with disease. We can associate those with the expression of other genes and see what they're linked to in the genome. And so that's what I did. I went into the databases and deciphered what other genes this gene was linked to, what other genes it was co-expressed with. And this is very solid data, actually, because it shows that this gene is linked to the expression of other genes, and it's very important. And in fact, this gene was linked to genes that are at the top of the pyramid of many important processes in the cell.
Now, after I discussed this paper on the show, we had one gentleman write in. He was very cordial, and I appreciate that, but he was skeptical of your work. Enrique's argument was that the gene encompassing the fusion site did not produce any known protein.
Well, my response to that is that we now know that there's over twice as many non-protein coding genes in the genome, which I just talked about, called long non-coding RNAs, that are highly important to the function of the genome. They're just as important as protein coding genes. And so his comments are made out of complete and total ignorance as to what and how the genome functions, because we now know that long non-coding RNAs and non-coding RNAs are the key to understanding genome function. It's like the protein coding genes are building blocks, and so these tend to be more similar among different creatures. But the non-coding parts of the genome that produce these non-coding RNAs are actually more organism specific, or what they call taxonomically restricted. And it's like these genes provide the metadata for the building blocks that are the protein coding genes to function. And they do a lot of different things. These non-coding RNA genes are found in the nucleus. The products of them, the RNA products of them, are found in the nucleus. They're found in the cytoplasm. Some of them are even exported outside the cell to other cells in structures called exosomes. So these non-coding RNAs do a whole plethora of things depending on the gene. And they're highly critical to the function of the cell. And like I said, this is the most important area of research right now in biomedical genomics that's going on, and it's receiving the most funding, and it's part of the ENCODE project, what we call the ENCODE project.
So they are critical life-supporting genes.
Yeah, they have knocked out these long non-coding RNA genes. If they have similar counterparts in mice, they have knocked them out and actually found that many are lethal, and that they're highly important. And in fact, many of them have been associated with all sorts of different types of cancer in humans.
More news has come out since the airing of these shows and our previous interview with Dr. Jeffrey Tomkins, who joins me again. He joins me from his office in Dallas. Welcome to the show again, Dr. Tomkins.
Thank you. Good to be back.
Now let's start off. I want to backtrack to a paper you came out with in 2011 addressing this claim that humans have a fused chromosome, which allegedly supports evolutionary claims.
Right. So Ken Miller actually gave an interview to a reporter that works for, what was that magazine? I've got it here, Mother Jones.
Oh, yes.
Magazine. Okay. Does that sound familiar?
Yes,
okay.
It does, yes.
So the reporter was a guy named Mooney. So Mooney basically got the interview
right.
from Ken Miller. So Ken Miller says that the fusion site is actually 1,300 bases away from the gene.
Right.
So a friend of mine at the Discovery Institute said, Jeff, did you see this article? And I said, no, I didn't. He said, well, this is what Miller is saying. He said, you need to contact him and get this thing settled. He said, you know, Ken Miller never responds to me whenever I send him an email, but maybe he'll respond to you. So I sent Ken Miller an email, and he actually responded within an hour. Very quick. And he was very cordial. He gave a nice long email explaining his position. But the interesting thing was he obviously didn't read my paper, because if he would have read my paper in its entirety, he wouldn't have said the things that he said. So I basically had to explain my entire paper to him over a course of several emails. And then he finally realized that he had made a mistake and that I was right. And so he said in the last email, talking about the long transcript, not the short transcript, or the long messenger RNA produced from this gene that has all 3 exons. He says, in this transcript, the fusion site is in the middle of the first exon, as you know. Well, actually, it's not in the middle of the first exon. It's in the middle of the first intron. But anyways, he acknowledged I was right. And then Casey Luskin actually took this piece from this email where Miller acknowledged I was right and published it in an article in The Blaze, which is an online media operation. And so people can actually get this article that Casey Luskin published with Ken Miller's quote from the email admitting I was right. So basically, everything is exactly as I described it, but not as I'm describing it really, but it's because that's what the data says. Well, this gene does not produce a protein. So there's over twice as many genes in the human genome as there are protein coding genes that produce RNAs. And these RNAs do all sorts of things. Some of them stay in the nucleus, some of them go out into the cytoplasm. A very small percentage are even used as signaling molecules in what are called exosomes outside the cell. But a lot of these long non-coding RNAs function in the nucleus, and they modify DNA. They're called chromatin modifying RNAs. And then a lot of them also combine with proteins to form these big complexes with proteins. The RNAs sort of act as backbones for these proteins to bind onto, and they also contain regions that bind to DNA as well in the genome. In fact, there was a paper a few years back where this particular secular research paper said that the genome is like an RNA machine. So really, it's an RNA world in the nucleus of a cell in the genome. And so this gene produces long non-coding RNAs of various different types. It's alternatively spliced. It has dual promoters. It has a promoter in front of the gene. Promoters are like switches in genes that turn the genes off and on and are involved in causing these genes to produce transcripts or RNAs at certain levels and certain amounts at certain rates in the right tissues at the right time. And so it turns out that the second promoter in this particular gene, it's called the DDX11L2 gene. The second promoter in this gene is the fusion site. So this is not just DNA sitting there doing nothing. The fusion site, and I have to use the term fusion site because it's really not a fusion site. It's a promoter inside a gene. And it's very common for genes to have promoters in their first intron inside them, as well as promoters in front of the gene. This is common across the human genome. And another important thing to understand is that you have genes on both strands of the DNA molecule. DNA is double-stranded. You have a plus strand and a minus strand. When we annotate genomes, we always annotate genes as to whether they're on the plus strand or the minus strand, and the genes run in opposite directions from each other on these strands. This particular gene is on the minus strand. So the message is not your typical message that's portrayed in the fusion site sequence. It's actually in the reverse complement of that sequence. So that's what the machinery in the nucleus, the various proteins that are called transcription factors are recognizing is in this other orientation on the minus strand. And so my new research that I have developed totally confirms that this fusion site is totally functional. It binds all sorts of transcription factors, over 50 different transcription factors, including RNA polymerase II, which is the main polymerase that transcribes genes in the genome. Okay, RNA polymerase II transcribes all the protein coding genes and the long non-coding RNA genes. And in fact, these genes, these protein coding genes and long non-coding RNA genes share many of the same transcription factors. But what's really cool about this gene that contains this alleged fusion site that functions as a second promoter is that it's highly networked with many other genes that are involved in high-level cell processes, high-level, very important cell processes. And so it's heavily networked with these other genes. It's heavily integrated into the genomic networks of the cell. It functions in over 250 different types of cell types that have been analyzed so far. And what's also really interesting is that my new research, mining the new ENCODE databases and these databases, this new version of a database in Japan called the Phantom database, which is also a database that studies gene expression, shows that this fusion site region, which is about 798 bases long, contains multiple start points for transcription where RNA polymerase II binds. So there's multiple transcription start points within this region, or transcription start sites. They call them TSSs for transcription start sites. So this is a hotbed of activity. It's not a fusion site. It's a functional region inside a gene that is completely on fire and highly expressed.
Right. And just for the sake of the viewers, when you say a transcription site or a starting site, you're saying there's multiple ones, basically, when it's reading the DNA and doing something with it?
That's where the messenger RNA is starting. The 5' end of that messenger RNA, that's where it is starting at the transcription start site.
Right. So you got multiple things that are starting in that region.
Yes.
All right.
Right.
Okay, well, thanks so much for taking the time to be on the show today. I know the viewers appreciate it.
All right, thank you.